Hey everyone, my name is Quinta Daniel, and I'll be talking to you guys about the characterization of transgenic tobacco constitutively expressing Galdieria sulphuraria superoxide dismutase, which is also known as GsSOD. As you guys may know, plants are severely impacted by climate change due to abiotic stressors like increased heat, salinity, and light, which induce excessive amounts of radical oxygen species. So really, this research aims to express SOD from Galdieria sulphuraria in tobacco to study whether it improves heat stress tolerance. So GsSOD was codon optimized and expressed in transgenic tobacco under the control of the HVHSP promoter. However, the promoter wasn't as effective as expected, rather causing majority of the plants to die, and some were actually contaminated along the way. So the procedure was then replicated with the 35S promoter using agrobacterium-mediated transformation and creating T0 lines. The transgenic lines were expected to be more resilient to higher temperatures and other abiotic stresses. So basically, we're trying to see if the novel traits demonstrated in model tobacco can be transferred to other economically important crops. So we did a number of procedures for our experiment. We conducted a seed sterilization and placed the plates in the refrigerator, in the growth chamber, and the heat chamber as well. We saw that the plates did not hold up well to the heat treatment, therefore we switched out the promoter, and these new plates were never heat treated and instead were later transplanted in the greenhouse. We also conducted DNA purification, in which we measured the concentrations of nucleic acids, and we also calculated the absorbance with the help of the nanodrop. Restriction digest was also prepared. In addition, we used PCR to see if the genes were amplified. Lastly, we used gel electrophoresis to see whether or not the genes were inserted in the tobacco's plant genome. In our table, we can see the concentration and purities of lines 1 through 3. Line 1 had a concentration of 17.6 with a purity of 2.08. Line 2 had a concentration of 26.1 and a purity of 1.78. And line 3 had a concentration of only 4 with a purity of 1.76. We can see that our concentrations are fairly small, especially line 3, and our purity the ideal range should be between 1.8 and 1.9, and lines 2 and 3 are around that area with 1.7 and 1.76. However, line 1 had a significant difference with a purity of 2.08. We also have a picture showing the gel electrophoresis results. There's a true band shown in line 1. We do see some band-like markings on line 2. However, this is not a true band. So the presence of a band indicates that the GsSOD gene was inserted into the plant genome of tobacco. So that is only shown for line 1. The initial sample collected for purification could have possibly affected the concentration of DNA found and the lack of the band in the GsSOD lines 2 and 3, especially line 3. And this is possibly because the plants were not put in the greenhouse, at least the GsSOD plant was not. And so the plants were rather small, so there wasn't much DNA that could have been extracted for the purification. And then the purity of line 1 could be high because of RNA contamination, and the bright band-like marking that was shown on the line 2 in the gel electrophoresis picture could indicate primer dimer, and primer dimer is just a byproduct of PCR, which causes the primer to anneal to themselves rather than the template. And this could be caused by poor PCR conditions like annealing temperature and the contamination of the template. So furthermore, in order for this experiment to possibly go well, samples from the GsSOD T0 lines should be recollected in order for DNA purification to be simulated once more. The T0 lines should also be placed in the greenhouse, and we should wait until the plants are further developed in order to get a better concentration. Also, DNA purification should be done carefully so that we can see results that will indicate our sample is pure. And so if all goes well, there should be bands present in all 3 lines, which would signify that the GsSOD gene was successfully inserted into the tobacco genome. Thanks.